Hello, everybody. Welcome to Miss McGuire video lectures. And today we are covering the cardiovascular system and specifically blood. And this is part one, where we cover only anatomy part. And I'm using OpenStax Anatomy and Physiology textbook. So let's go ahead and begin. So functions of blood, transportation. Your blood will transfer nutrients, oxygen, hormones, waste product, defense, like your white blood cells will defend you from pathogens. They can defend you from your own tumor cells. And clotting is also defense because that will protect you from blood loss. So your blood will clot to prevent bleeding, external or internal bleeding, and that will save your life. So that's defense. Homeostasis is also a function of blood. Your blood is involved in the temperature regulation and the chemical balance of your body. Now, what is your blood made of? So your blood is connective tissue, and it's made of cells and extracellular matrix. Specifically, when we're talking about blood, the extracellular matrix is plasma. So the blood is made of plasma and what we call formed elements. We don't call them cells because some of them are not complete cells, right? And we will look at it in a minute. But anyway, your blood is made of plasma, that mostly water. Plasma has proteins, nutrients, hormones, and cells, white blood cells, red blood cells, and also platelets. Right now, if you draw a patient blood and you put it in a test tube and you centrifuge it, so what centrifuge gonna do, it's gonna separate blood into several layers. The more heavier part, red blood cells will be on the bottom, then the light part like plasma will be on the top, and between them you will have buffy coat that composed of white blood cells and platelets. Right now, what is hematocrit? Hematocrit is a percentage of RBC or red blood cells within this sample of blood. Right, so whatever settles on the bottom of the test tube will be hematocrit. And normal blood hematocrit is about 45% of whole blood. So 45% hematocrit, and then we have 55% of plasma and white blood cells and platelets. So you can see the hematocrit, it's a little bit higher in males and a little bit lower in females. Right, this is what it says. The buffy coat will include WBC or white blood cells and platelets. Hematocrit will be on the bottom, and plasma include water, proteins, nutrients, and hormones. And you can see when we have decreased hematocrit, that indicates anemia in a patient. If we have elevated hematocrit, then it's indicate polycythemia in patients. So too much of hematocrit or too little. Oh, I apologize. So general characteristic of blood, it's a viscous fluid about 38 degrees of Celsius. pH is 7.35, 7.45. We have about 5 liters of blood, a little bit more in males, a little bit less in females, but again, the average is 5 liters. Now, plasma is 92% water and has plasma proteins and other solutes. Now, if you look at this table over here, the only thing that is not correct here is actually this line, because so you see plasma and you see formed elements, but erythrocytes or red blood cells, they are part of formed elements. So we need line over here. All right, so you see plasma, 92% of water, plasma proteins about 7%. Then we have regulatory proteins less than 1%, and other solutes such as nutrients, gases, waste, that is about 1%. And then we have formed elements, including erythrocytes, right? 99% of these formed elements, and leukocytes and platelets over here, right? Now, most of the plasma proteins are synthesized by liver. Formed elements are formed within red bone marrow. And formation of blood formed elements or blood cells, including RBCs, WBC, and platelets, is called hematopoiesis. Now, all these cells are short-lived. So they live, like RBCs live about 4 months, and white blood cells live sometimes just a few hours, right? So that's why we have constant production of blood cells. Now, sites of hematopoiesis changes over time, right? During embryonic development, most of the blood cells are synthesized inside yolk sac, they in liver, in spleen, but you can see all these declines, right? So that's our birth right here. And before birth, we have hematopoiesis, prenatal hematopoiesis in yolk sac, liver, and spleen. And then you see the hematopoiesis decreases completely to a zero. And we start producing blood cells in the bone marrow, right? And again, in the bone marrow, we have different bones like tibia and femur. Those are long bones. This production within those bones are also declines. So when we adult, we pretty much have hematopoiesis in vertebra and pelvic bones. Right. In a lymph node over here, well, all blood cells are formed within bone marrow. Then some of them, like lymphocytes, they will migrate into lymph nodes and they will reside over there, and they will be capable of dividing and proliferation within lymph nodes. So that stays pretty much constant. But that's only for white blood cells, not for red blood cells, right? And again, they form within bone marrows and they migrate. Lymphocytes migrate within lymph nodes. Or they can migrate also within thymus, that is not shown over here. And so T cells will mature in the thymus, B cells will mature in a lymph nodes. Now, all formed elements come from the same progenitor cells. It's stem cell that is multipotent. So it's a multipotent hematopoietic stem cells, a hematoblast. And some of this, so this stem cell will divide, and some cells after division remain stem cells and other will differentiate. So this cell can differentiate either in myeloid or lymphoid stem cell. And lymphoid stem cell will differentiate into natural killer cells, T cells, and B cells. So those are lymphocytes over here, right? So lymphoblast, lymphocytes, and natural killer cell is also large granular lymphocytes, right? Myeloid stem cells will differentiate into erythrocytes, red blood cells, into rest of white blood cells, basophil, neutrophils, eosinophils, monocytes. Monocytes will become macrophages, right, in future, and also into megakaryocytes that will become platelets. So you can see either those will be lymphoid cells or they're gonna be the rest of the cells, including white blood cells, red blood cells, and platelets. Now for this differentiation and for hematopoiesis, we will need hematopoietic growth factors like erythropoietin, synthesized by your kidneys. It's a glycoprotein secreted by kidneys, increased RBC synthesis and production. Thrombopoietin, also synthesized by kidneys and liver, and it's also glycoprotein, important for development of platelets. And then we have cytokines. So those are several factors, several autocrine, paracrine hormones that are necessary to stimulate progenitor cells and also stimulate immune response. So those growth factors mostly secreted by kidneys and the rest of them by liver, essential for differentiation of the cells, right, that we just discussed in our previous slide. Now, this is a summary of formed elements, and you can see erythrocytes, leukocytes, including lymphocytes over here, right? But I will not go, I'm not going over this table. Please stop the video and just read it. And, you know, it's all self-explanatory. Plus, we will cover this in, you know, in our next slides. Anyway, I just want to kind of look at several things over here, like erythrocytes or red blood cells. Look, we have in a microliter, right? We have more than 5 million, about 5.5 million of red blood cells in 1 microliter of blood, right? So that's really amazing. Erythrocytes are one of the smallest cells in your body. Now, if you compare them with, for example, leukocytes, the total of leukocytes, right? White blood cells, we have 7,000 only, right? So we have way, way more. We're talking about millions here, right? Or 7,000 of white blood cells. And we have about 350,000 platelets, but still considerably smaller amount compared to red blood cells. Now, structure of erythrocytes. Erythrocytes are not complete cells. That's why we call all cells that, you know, part of your blood formed elements. Like erythrocytes, not complete cells. Platelets, fragments of cells. The only complete cell are your white blood cells, leukocytes. Erythrocytes, they do not have nucleus, and many organelles are missing, like mitochondria. They also lack endoplasmic reticulum. And erythrocytes gonna lose nucleus and most of its organelle to increase this volume and space to carry hemoglobin inside, and hemoglobin will carry oxygen, right? So more space for oxygen, and you don't need mitochondria because mitochondria can use this oxygen for cellular respiration. So they're gonna make ATP by without mitochondria. So it's gonna be glycolysis, by glycolysis. So erythrocytes biconcave disc that also increase surface area, and structural proteins like spectrin are flexible. And on this diagram, you can see that you know, development of erythrocytes, and we start with pro-erythroblast, then it became erythroblast, then normoblast, and this normoblast gonna eject the nucleus, eject most of the organelles, right, and became a reticulocyte. Reticulocytes are released in the bloodstream, and they mature into red blood cells. Now the main okay, so we don't have nucleus, we don't have mitochondria, we don't have ER. So the most of the space within erythrocytes are occupied by hemoglobin. Hemoglobin is the globular protein. It's made of 4 polypeptides. So if you see over here, kind of this is one molecule of hemoglobin, and it's represented by 4 colors, right? So this is 4. We have 4 polypeptides forming the quaternary structure of hemoglobin molecule. Each globin binds to a heme group. So here we have a heme group, and inside this heme group, we will have iron. So you can, I'm sorry, you can see this molecule over here. That's a heme, and inside the iron. And each ion can bind to 1 oxygen. Now, when oxygen binds to hemoglobin, we call it oxyhemoglobin. So hemoglobin picks up oxygen in lungs. When hemoglobin release oxygen in your tissue, we call it deoxyhemoglobin. And some carbon dioxide can bind to hemoglobin as well. Then we call it carbaminohemoglobin. So the erythrocytes live about 4 months, 120 days, right? And they are produced within your bone marrow, and then they removed from circulation when they, after their lifespan by macrophages, and it happened in your spleen, in your liver, or in your bone marrow as well, right? 2 million cells produced per second. That's amazing. And we need iron, copper, zinc, B vitamins for normal production of RBCs. So on this diagram, I know my picture is on your way. But you can see over here, hematopoiesis begins in the bone marrow, right? We go from stem cells to erythroblast, reticulocyte, and then reticulocytes mature into red blood cells. And then red blood cells, carry on their function, like carry oxygen, right? Carry some carbon dioxide, because some carbon dioxide carried within plasma, but some carried within RBCs, right? Back to your lungs to release. So they just continue doing their regular job for 120 days approximately. And when they get damaged and old, they will be phagocytized by macrophages in the bone marrow, liver, and spleen. And we're going to recycle pretty much everything, right? So, so you can see over here macrophage engulfing RBCs, and then this phagosome gonna fuse with lysosome. Lysosome releases digestive enzyme and pretty much break down these old damaged cells. And because we have so much hemoglobin, what are we gonna do with that? Well, hemoglobin will be split into heme, right, and globin. Well, globin is a protein. Protein is made of amino acids, so that's gonna be broken down into amino acid, and they can be reused, right, for new synthesis or for other needs of your cells whenever they need amino acids. Now the heme will split into iron and heme group, and the heme group will be converted into biliverdin and will be stored inside your liver, or it can be converted into bilirubin in your liver. So it will be stored in your liver and used for hematopoiesis, or it can be turned into bilirubin and be used to make bile by liver. So your liver make bile using this heme group. Iron will be attached to a special carrier, protein carrier, transferrin. Trans means you're gonna transport something, and ferrin is a ferrum, that's an iron. That's the name of this, transferrin. And it's also can be stored in the liver. And when it's needed, it's moves, through your bloodstream, obviously, inside your bone marrow, and we start, hematopoiesis again, right? So that's a life cycle of erythrocytes. So disorders of erythrocytes. So it can be wrong size, wrong shape, number of erythrocytes, so everything matters. So anemia is kind of umbrella term for 400 different type of diseases. Anemia just means reduced capacity of your blood to carry oxygen. But why your blood is not carrying enough oxygen, that can be caused by different causes. For example, it can be deficiency of RBCs, or it can be low hemoglobin number, or it can be caused by blood loss, or faulty RBC production, or disruption of RBCs, right? So all, 400 types, you can pretty much think about any possible cause over here. One example given here is sickle cell disease. That's a genetic disorder, and hemoglobin as a protein doesn't have its normal shape. And when protein is misshaped, the whole cells are misshaped. So you can see over here, sickle cell, red blood cells, right? And that delivers less oxygen. So deliver less oxygen, that's anemia. It can be iron deficiency anemia, the most common type of anemia, vitamin B12, or folate deficiency anemia, or, over here you can see like that's a normal blood, microcytic, macrocytic, hypochronic anemia. So the, RBCs can be small or large, or they do not have enough hemoglobin, right? A deficiency of vitamins, so on. Polycythemia is opposite. When we have elevated RBC count, it increases viscosity of blood and make heart difficult to pump blood. Right, so that's a disorders. Now, leukocytes characteristic. They are less numerous than RBCs. So we have about 5,000 to 5 to 1,000 per microliter, right? So we can just remember, I think the first one was better, like 4,000 per microliter. They are larger than RBCs. They are complete cells, but they have an even shorter lifespans, a few hours, even few minutes. So on this diagram, you can see that leukocytes can be divided into granulocytes and agranulocytes. A granulocyte means when we use a special technique to dye them, we will see granules is inside the plasma, or we will not see granules, right? And then will be a granulocyte. I think the next one, oh no. Okay, so, oops, sorry. So granulocytes will include neutrophil, eosinophil, basophil, and agranulocyte include lymphocytes and monocytes, right? So it will be some other slides about it. Leukocyte characteristic, those are the cells that can move. Very few cells in your body can move. Actually, only leukocytes and sperm cells. All other cells, they cannot move on its own. You would think my red blood cells move, but your red blood cells cannot move if your heart doesn't pump your blood. So red blood cells move within your blood, but they cannot move on its own. For example, ovum or egg cannot move on its own, right? So that is two cells, leukocytes and sperm. Here are the processes here, let's see. And again, I try to move my cell away, but I don't think it works. But when you have an injury, right, those cells that are injured and bacteria that, you know, invade your wound, they will secrete chemicals. And those chemicals will attract leukocytes, right? And this is called positive chemotaxis. So it's over here, positive chemotaxis. So they will be attracted to the site of the injury, and they will squeeze between the cell of a capillary wall and follow these chemical signals, right? And well, some of them, let's say monocytes, they will differentiate into macrophages and they will phagocytize the pathogens, but it will be also, let's say, what it says here, neutrophils, right? Neutrophils are called microphages. So we have macrophages, and those are microphages because they actually also capable of phagocytosis, the same as the same as all of them are like eosinophils, neutrophils. Eosinophils and neutrophils are both capable of phagocytosis, right? And plus, they can release chemicals that, you know, it says here break down pathogens, but that's, I doubt very much. But anyway, when we have our video about immune system, we will talk into more details of what neutrophils do, right, and how they different from macrophages. But over here, we just want to know that those leukocytes will migrate to the site of injury by diaperesis, so they will move like amoeba, and they can squeeze between the cells of capillary wall, and they're gonna do their job engulfing and neutralizing the pathogens. Okay, so here we have our finally classification. So we have granular leukocytes, they contains granules and include neutrophils, eosinophils, and basophils. Neutrophils most common, about 50–70% of leukocytes. They are polymorphonuclear, or we call them polys. And this is because, look over here, that's a nucleus. Isn't it amazing? One nucleus and it has several lobes, so many lobes. Polymorphonuclear. So it has many lobes of the nucleus, or just polys, and they are phagocytes. Eosinophils, they will have, so because they're most common, right? Do you know what target is? The target are bacteria, but because bacteria is pretty much our main infection agent. So they will be effective against bacteria. Eosinophils, effective with parasitic worms. You also will see increase of eosinophils in a patient with allergy. So that's why they produce antihistamine. It's anti-inflammatory substance, and they will be like really pink, red color. I mean, it's not natural color. We're gonna use dye. So under microscope, you will see them pink, and the nucleus has two lobes. So that's remind me sunglasses. So, and basophils, they least common, and they release histamine, and histamine is inflammatory chemical, and it will initiate, along with other inflammatory chemicals, initiate inflammatory response. Then we have agranulocytes. They have less visible granules. Lymphocytes, this is the one that will include B cells and T cells and natural killer cells. B cells secrete antibodies, and T cells are important in cell-mediated immunity. So those are really important cells. Monocytes, they are phagocytic, and they will be monocytes when they're in the bloodstream, but when they enter tissue, we call them macrophages, right? So now if to remember, relative concentration of all these leukocytes, there is a mnemonic. Never let monkeys eat bananas. So neutrophils, we have they're more numerous, right? So here's our neutrophils, 50 to 70%. Then we have lymphocytes, right? Then we have monocytes, eosinophils, and basophils is the least common leukocyte. So never let monkeys eat bananas. And monocytes, they're really huge cells. So you can see how big the cell is compared with red blood cells. And lymphocytes, so monsters, big, and lymphocytes are the smallest of all leukocytes. And it's almost has like so dark cytoplasm that you cannot even see nucleus. Leukopenia is a disorder, too few leukocytes, leukemia, abundance of leukocytes, and it's a cancer, and lymphoma is malignant tumor. T or B lymphocytes, accumulate in lymph nodes. So, okay. Platelets. So another name for platelets is thrombocytes. They are, small fragments of cells. So we have good amount of them, right? 150 to 160,000. And they have 10-day lifespan in the blood. They will migrate to spleen, and the cells that will, so first the megakaryocyte is formed, and then megakaryocyte will be pretty much broken into platelets. So it says release platelets over here. But yeah, platelets are fragments of megakaryocytes. And on a blood smear, you can see this small, but very, very critical part of your blood platelets. So they important in a hemostasis, so stoppage of blood bleeding, right? So clinical function, hemostasis, and also they secrete growth factors. So you can see very quickly here, just a small slide, how we have our myeloid stem cells, and it's differentiate into megakaryoblast. We do need thrombopoietin from the liver and kidneys, right, to initiate this process, and those are hormones, right? So megakaryoblast become megakaryocytes, and this is a precursor of the platelets. So you see like this little pieces breaks off and became platelets. Thrombocytosis is a condition when it's too many platelets, and thrombocytopenia, too few platelets, right? And neither one is good, because thrombocytosis, if you have too many platelets, then you can form unwanted clots, and thrombocytopenia, clot is not formed when you need it. Okay, so we're doing good over here. I'm trying to make this video not that long. So the last topic covered in this video lecture is blood type, ABO and RH groups. So if you look at red blood cells, they have antigens on the surface of their cell membrane. So more than 50 antigens identified. but most significant are ABO and RH. And that's why we have blood of different group. So we have blood A, blood B, blood AB, or O. And the RH will tell us is it positive or negative blood type, right? So now look, that's show us the red blood cells. So this one has antigens, group A antigens, right? This has a special antigen B, this has both A and B, and this has none. Now, if you have antigens on the surface of red blood cells inside plasma, so that's a plasma, you will have antibodies. So if you have antigens A, right, you in your plasma, you have anti-B antibodies. So your plasma is ready to attack blood B, right? You don't want to attack blood A, but you're ready, you have weapon, anti-B. It's against B, right? Anti-B. So you have this weapon to fight group B. If you type B, right, then what? You prepare to fight antigens A. So you have anti-A. So you have this weapon. So if somebody's blood enters your bloodstream, you have your antibodies, all right? If you AB, everything is yours. A is my antigen, B is my antigen. So I don't have any antibodies. I'm welcoming all type of blood, right? If you O, or you know, for you don't have any antibodies. Oh, I'm sorry, you don't have any antigens, no antigens. So you prepared to fight all type of blood. You have anti-A and you have anti-B. So kind of remember, antibodies is a part of your immune system. So your immune system is designed to protect you from foreign substances. If I am type A blood, right, then type B is a foreign substance for me. If I am type B blood, type A is a foreign substance for me. That's why my immune system has these antibodies ready for me. If I'm O, then I don't have any antigens, right? No, none are antigens. So everything is foreign for me. A is foreign, B is foreign, AB is foreign, and so on, right? That makes sense? Okay, so that's how we define the groups. Depends of what antigens are present, we define a group type. And then for this RH blood group, we have RH antigens, dozen of RH antigens identify, and only D is clinically relevant. So if I have this D antigen, I will be blood positive, right? HR positive. If I lack of D antigen, I will be RH negative, right? And then if I am RH positive, I don't have any antibodies against this D antigen, anti-D. I don't have it because I do have this antigen. But if I RH negative, I will produce antibodies against this antigen. So I pretty much gonna destroy this antigen, right? So you can see here an interesting summary that you would think that, okay, so the most common blood type is O positive over here, right? So you see, you know, pretty much very common in all, you know, groups. Okay. Now, this slide explains us what is hemolytic disease of a newborn. This happens when mother is Rh negative and baby is Rh positive. So because baby is Rh positive, right, it has D, so that's a red blood cell of baby, right? That's a baby. And it will have antigens D. So that's gonna be D. So baby will have it, D. So mother gonna produce antibodies against this D, right? And especially this is dangerous in the second pregnancy, because mother already will develop large amount of antibodies, and those antibodies can cross the placenta and enter the baby bloodstream, and it's gonna attack the embryo, and it can cause anemia and death of the fetus. And of course, we have some medication now that prevents development of antibodies, Rh antibodies in a Rh negative mother, right? So, and again, you can stop over here and read this, diagram. Those diagrams have very, very important information. But the point is, mother start producing antibodies that will attack as a fetus red blood cells. Okay, and the last slide here, how we determine, we just have kind of 2 more slides, but here how we can determine the blood type. So this is how the procedure works. So you have unknown blood samples is put into wells. So here sample 1, right, 1, 2, 3, and 4, and then you're gonna add. So you put blood 1 in 1, 2, 3, 4 wells, right? So you put blood in 4 wells. You put sample 2 in 4 wells, and 3 and 4, and then you're gonna add anti-A in this well, anti-B, and anti-D. So if you have here A, and you add anti-A, so antibody gonna binds to antigen, and blood will clot over here. So you will see this agglutination of the blood. Sorry, not clot, that's not correct. It's called agglutination. And if you have D over here, right, so here's a D. When you add anti-D, you will see agglutination as well. So this person is A positive. So this person is B positive. This person is AB positive. And this, we don't have agglutination in any of them. And then this is O negative. Now, when I told you coagulation, again, I just want to make it sure, coagulation is not what agglutination is. Coagulation require lots of special coagulating proteins. It's a very complicated process, and this is how you stop bleeding. Agglutination includes antigens and antibodies. That's what agglutination is, right? And why we have universal donor and universal recipient? Well, obviously, universal recipient should be a person that's not gonna fight any of other blood types. So who would never fight any other blood? That's gonna be person AB positive, because you already, everything your immune system recognize as yourself, like antigen B and antigen A, and if you positive, antigen D. So your immune system gonna recognize everything as your own. So you don't have any antibodies in the plasma, right? None. So whatever going to your plasma, you're not gonna attack it. So you don't have the weapon to fight it. So that's gonna be our universal recipient. Now, universal donor would be a person who doesn't have any antigen. So you can give this blood to anybody, right? Because you give me this blood and I am B positive, right? So I'm B positive, so I have anti-A. right? So you give me this blood, right? And it doesn't have any antigens A, so I'm not gonna fight it, right? The person with type B, it have anti-B, but this doesn't have any B antigen, so it's not gonna fight it either. So this is our universal donor, and this is our universal recipient, right? But this universal donor, it can donate blood to everyone, but it only accepts blood from a person type O, right? Because it has anti-A and anti-B. So you give this blood to this person, look, I have these antibodies to fight this blood type. You give me this blood, I have this one to fight you. You give me this blood, I have this one to fight you, right? This antibodies. So this will be universal donor, but when person with blood type O need a blood transfusion, we can only give him or her the same blood type O, right? So makes sense? Okay, so so that was our last slide for today. I will record another video on physiology and we will talk about hemostasis. But if you are in my anatomy class, that's all you need to know, right? And don't forget to read your textbook. That's important too. So thank you for watching and I hope it was helpful.